Mutant ZP1 in familial infertility. Definition: Zona pellucida sperm-binding protein 1 is a protein that cross-links CP2 and CP3. Aberrant CP1 results in sequestration of CP3 in the cytoplasm, thereby preventing the formation of the zona pellucida around the oocyte.
When ejaculation occurs during intercourse, approximately 200 million sperm, or spermatozoa, are deposited into the vagina. They swim through the cervix, propelled by whip-like motions of their tails, or flagella, after which muscular contractions of the uterus direct them to the uterine tubes. This process usually takes between 30 minutes and 2 hours. Only around 200 spermatozoa will reach the secondary oocyte in the uterus.
Application in medicine. It's vital for the production of oocytes in early development, for fertilization, and for protection of early embryos before implantation. In mice, a target knockout of either the gene encoding CP2 or the gene encoding CP3 precludes the development of a zona pellucida, leading to a sterility. However, to our knowledge, previous study of defects in the zona pellucida in humans have not identified mutations in any of the 4 genes, CP1, CP2, CP3, or CP4, encoding the zona pellucida glycoproteins, which are secreted by growing oocytes. Current models of the development of the zona pellucida involve the independent synthesis and transport of each zona pellucida protein through the endomembrane system toward the cell surface. After the cleavage of C-terminal domains near the plasma membrane, the zona pellucida proteins are released into the extracellular space, repeating zona pellucida proteins from long filaments, which are cross-linked probably by CP1, to form the zona pellucida.
To know is that when they're considering getting pregnant, they should consult their family doctor or gynecologist for preconception counseling. That means going in before they actually get pregnant to determine if there are any changes in their medications that they need, or if there are any medical problems that might complicate their pregnancy, and if there are any genetic mutations that they're at risk for. And the gynecologist will go over all those things and give them the go-ahead. Start her on vitamins, on folic acid.
Oh, so you should be taking vitamins even before you conceive?
Yes.
Pros and cons.
Okay, so this segment now is going to focus on fertilization, implantation, and pregnancy. What we're going to cover in this series of notes is looking at the events of fertilization, then we'll talk about early embryonic development and implantation, as well as embryonic and fetal development, and then hormonal changes in pregnancy. So we're going to be covering all 4 topics. So in fertilization usually, but usually because that's what the normal is, it's normal for it to take place in the uterine tube.
Pros: we detect an autosomal recessive pattern of inheritance for homozygous deletion in CP1 in certain members of a family affected by infertility. Our data suggests that the mutation prevents the formation of the zona pellucida around the ova. All observed oocytes from affected family members lack a zona pellucida when examined with light microscopy. In oocytes from family members IV5, we did not observe the expression of CP1 and CP3 in the region normally occupied by the zona matrix. We therefore conclude that oocytes from affected family members lack the zona matrix, which account for the infertility. Cons: the conservation between the 1390FX4 F404X mutation in ZP1 and infertility supports but does not prove causality. Non-mutant ZP1 contains 638 amino acids, of which 270 make up the zona pellucida domain, a protein aggregation module spinning amino acids, 279, 21. The mutation is predicted to disrupt this domain and yet permit interaction between the mutant ZP1 protein and the other zona pellucida proteins, ZP2, ZP3, and ZP4. The loss of C-terminal domains in the mutant ZP1, however, will be expected to upregulate essential normal functions, such as C-terminal cleavage, transmembrane location, assembly, prevention of intracellular polymerization, and trafficking of the protein both intracellularly and the exterior of the ovum. Issue apply in Ecuador. Female infertility varies widely by geographic location around the world. In 2010, the countries with the lowest rates of female infertility include the South American countries of Peru, Ecuador, and Bolivia. Historical and regional insight about Ecuador, specifically with regard to inequitable resource distribution, allow for a better understanding of why the arrival of assisted reproduction treatment in Ecuador brought few of the anxieties about nature that had been common in the global north concerning different ontological orders. For instance, concerning its artificiality.
I'm Dr. Patricia Nan. I'm an OBGYN at Mercy West in Janesville. I think of myself basically as a problem solver, and it marries those two up very nicely. One of my favorite parts of obstetrics is that moment when the mom delivers and you hand the baby to the mom and the dad. There is such a transformation that takes place there, and it's just beautiful to see, and I'm honored to be a part of that. What I love most about my job is the people, and just helping people sort through and make sense and make a plan and get better and feel better. Overall, I'm there. It's their visit, not mine. And everything that we do should be geared toward the patient. One day, almost everybody.
Conclusion. We propose that the difference between the oocytes of mice lacking ZP1 and those of the affected woman homozygous for the truncating ZP1 mutation described here is explained by the retention of the partial zona pellucida domain and the absence of C-terminal domains in the mutant protein that promotes interaction with and sequestration of other zona pellucida proteins. The intracellular sequestration of these proteins could impede their trafficking to the extracellular milieu, thus precluding the formation of a zona pellucida and leading to infertility. The amount of truncated ZP1 protein in heterozygous subjects, in contrast, may be insufficient to sequester other zona pellucida protein to the extent that the zona pellucida is functionally compromised. The observation in an affected sister, IV5, of the apparent absence of ZP1 and ZP3 surrounding X and the collocation of ZP1 and ZP3 inside the X support our hypothesis. Thank you.